Good morning, everyone. Welcome to day 2 of the GYAN course. So yesterday we covered a few concepts on radiology informatics, where we covered, basics of radiomic feature extraction and how to use some of the more advanced concepts, more context-driven approaches for radiology AI, including human in the loop or radiologist in the loop AI systems. So if you have any questions regarding that, we can always discuss after this lecture. But today I want to switch gears and talk about AI in digital pathology. So how many of you here are, let's say, familiar to some extent with what digital pathology is? I know that I briefly introduced it yesterday, but any of you here have worked with digital pathology? So what comes to your mind when you hear the words digital pathology?
Glucometer, sir. Glucometer pathology. We are testing the sugar test now, some morning. So that is digital
No,
pathology. I'm thinking.
it's not that.
Yes.
So let's have the mic here.
Sir, I think using AI to detect some kind of disease pathologically.
So I think that's the general theme of this entire series, right? So we are using AI to do something to detect disease and prognosis and whatnot. But what comes to mind when you hear about digital pathology? So maybe we can have the mic here.
So in traditional we have this slide mechanism where you have slide, you scan it, but in
Right.
digital we have a whole slide imaging, so that we take the scan of entire slides and later process using some techniques and diagnose and pre-process those things.
Right. So what happens traditionally is, so if there's some, let's say, suspected nodule or suspected lesion, what happens is the surgeon would go in, resect or take a biopsy of that particular region, and then they would take it out, mount it on a glass slide. They would fix it with different dyes, and then they would give it to a pathologist, and the pathologist can then review that slide under the microscope. So why does a pathologist do that? So if the pathologist does that, they can then look for any abnormalities, if there are any cancer cells or some other regions of interest that can point to some disease, right? So that's the traditional mechanism of how pathology works. Now in digital pathology, what happens is you take that slide and you digitize it, and then instead of the pathologist having to look at it under the microscope, they would look at the computer screen, look at the slide, look at the image, and do whatever analysis they have to do later on. So in the course of the next hour and a half, what we will do is we will cover some of the fundamentals of digital pathology, especially computational pathology. What are the advantages, how these can be used, and how in fact this is sort of a paradigm shift in the domain of pathology. So what is digital pathology? So like I said, it incorporates the acquisition, management, sharing, and interpretation of digitized slides. So all of these terms, acquisition, management, sharing, and interpretation, all of these are important because by combining all of these, we have essentially moved from a more traditional analysis of these pathology images to more modernized analysis, where we look at these images, or the pathologists look at these images on the screen. So starting from the process of acquisition, so how these are digitized, to management, because these are images, right, and these are huge images, so you need a lot of storage. And then sharing, so because these are images, you can share them, and then share them. So traditionally what happens is, if you have a slide, you would ship the slides, right? You would physically ship the slides. So there's no more shipping of the slides, no more couriering the slides. So you can actually send it via internet. And then interpretation. So this multiple pathologists can sit at the same time and they can interpret the same image at different locations. So digital slides are created when glass slides are captured with a scanning device, and we'll see what the scanning device looks like. What this does is it provides a high resolution digital image that can be viewed on a screen or a mobile device. So imagine, let's say a pathologist is traveling, right? So they're not at the desk, so they don't really have access to a microscope. But if the slide can be digitized and it can be transferred to their whatever server there is, they can, if there's a proper viewer, they can open it on their phone, on the smartphone, and can view it. So it enables something called telepathology. So you can capture the entire glass slide, and so we heard a couple minutes ago the term whole slide image. So that's when you capture the entire glass slide under bright field or fluorescent conditions at a magnification that is comparable to what you can actually observe under a microscope. And last but not the least, you can actually share it using specialized pathology software applications. Now, the buck doesn't really stop there. Yes, this process has been digitized, but then where can we come in? As imaging researchers, as scientists, where do we come in? So now that we have access to these digital images, we can apply our fancy AI tools, our mathematical models, and analyze these images in a way that's almost impossible by just looking at these images, similar to what we saw in radiology yesterday. So here's a summary. You have the traditional pathology workflow here, where you have the tissue preparation, and like I said, if you don't really have an expert in that physical location, you have to ship the slides to a lab where it is analyzed, and then you have a patient diagnosis. However, with digital pathology, after the tissue preparation, you have the step which creates the digital slides, and then everything is computerized after that. So you have the analysis of the digital slides. I mean, obviously you have the pathologist who can look at the digital slides, but then you can build your AI systems for automatically diagnosing diseases or doing whatever, let's say, a morphometric analysis that you have to do. So one of the first benefits of digital pathology is quality. So there are algorithms for analyzing slides, and these are objective, accurate, and quicker than microscopy. So imagine a pathologist has to look at a slide under the microscope, they would take some time. But if you can actually digitize it, so instead of spending time on getting the slide, mounting it on your microscope, and trying to move it, everything can be done with the click of a mouse, right? So, and there's rapid access to prior cases. When it comes to looking at a lot of times pathologists might be interested in looking at previous slides. If they want to look at previous slides, what they have to do is they have to, let's say, unmount the current slide, find out where in the rack the previous slide is, mount it, and then view it, right? So there is some time involved there. I'm not saying that it's a lot of time, but there's some time involved there, right? And these slides have to be stored. I mean, they have to be physically stored. When it comes to digital pathology, you basically store these slides digitally. So you can, at any point, if you know your folder structure, you can go back, you can access your previous slides, and you can look at them and you can analyze them. And the data storage allows for long-term predictive analysis, right? So a lot of times, let's say if you don't have enough physical storage space, or for some reason, the slides that have been there for 10 years, 15 years, there's a chance that the physical slides might get lost or might get damaged. But if you have them digitally, and if you have a backup of that on some server, you will have them theoretically forever, right? So let's say that you want to teach the pathologist wants to teach their residents or their fellows, their medical students. There are some interesting cases that they have observed 5 years ago, 10 years ago. They can keep a note of that on their server, and they can immediately pull that slide, as opposed to having it physically, where they have to store it somewhere, right? So that's how it becomes important, and the quality is also enhanced. Errors are reduced. So you have barcoding. So each of the slides has a separate barcode, and based on that barcode, you can pull whatever slide you want, and better views. So you can have zoom in and zoom out at multiple angles, and then you can measure multiple areas of interest. So if one area is important, they can go to that area, immediately zoom in, and they can on their software, they can mark that a particular area. So let's say that you have this whole slide image, glass slide, there's this whole slide image, and they're looking at it on a computer screen. So this region is important. They somehow they have some sort of marker and say that this is an important region, and it's a digitized marker, right? It's just a pen mark, so it stays there, and when they come back, they can always look at it, find out what it is that they annotated, and that can also be used for downstream analysis. Okay, so here's just a summary of what the text here says. You have improved analysis, better views, and it can reduce errors. So all of this is connected to the quality of the images that digital pathology helps us, or digitized pathology slides help us achieve. Now productivity, right? So this is also very important, because one of the main let's say driving factors of research is collaboration. So eventually we would want to collaborate with scientists from other fields. As a pathologist, they would want to collaborate with computer scientists or biomedical researchers to analyze these slides, right? So how do we share the slides? You can't expect to give this slide to a biomedical researcher. They look at it under the microscope, and there's not much that they can do. I mean, maybe they can do some qualitative analysis, but there's not much you can do beyond the qualitative analysis by looking at the slides under a microscope. But if the slides are digitized, you can share it with other people. We routinely have collaborators sharing slides with us. So we can take these images, we can look at it in multiple different ways, and then we can apply our deep learning techniques, our handcrafted feature analysis, and get information which otherwise would be impossible. So it allows for automation and work schedules, remote access. So let's say that, like I said, a pathologist might be traveling, they would want to access these slides remotely. So you can actually do that if you have these digitized slides. So reduced turnaround times, similar to what I discussed in the previous slide. It reduces because of digitization. It reduces time retrieving and data matching, organizing these slides, and speeds up access to samples and improves turnaround time. So why is this important? Because pathologists every day, they have to look at hundreds of slides, right? So just taking slides that are physically there, mounting it on the microscope, reading the slide, analyzing the slide, writing the reports, then putting it back, getting another slide. I mean, yes, traditionally pathologists are used to that, but imagine if something like this can be done digitally, it would definitely improve the turnaround time. But obviously, that doesn't mean that this change would happen overnight. So there has been a steep learning curve that has been observed when it came to digitizing these slides, and not just digitizing the slides, going from a traditional pathology workflow to a digitized workflow. So there are traditional pathologists who still resist it, because they're used to their previous protocols. Now there will obviously be a change in protocol, but everyone has to adapt, and slowly they have been adapting, and there are multiple hospitals over the world that have gone completely digital when it comes to pathology. And where it becomes very important for us is innovation. So eventually this is big data, right? So if we can digitize these slides, there's so much data, so much information that we can obtain from these images. So it allows pathologists to become more specialized. So let's say that a pathologist wants to completely focus on prostate cancer, or completely focus on breast cancer. So now they have, with digitization, they can actually spend more time on a particular field. They can spend more time doing research in that particular field, right? So it can also help us deliver better tools for teaching and training. So imagine there's a microscope, and I'm sure most of you here would have been exposed to some microscopes in your high school or in your college, junior college. So imagine that your teacher, your professor wants to show you something under the microscope. So they would adjust the slide, and they would ask you to come there and take a look and see, okay, you see this particular portion of the slide, this is what it means. Now, so just for a second, imagine if there were 10 students in the class, right? So the teacher would, or your professor would identify that area of interest, and one by one they would ask each of you to come and observe. And this is what happened when I was in +2, and then I was doing some, I think they were looking at some, I don't know, it was some seed or something that in our botany class, I remember, right? So now if it was digitized, let's say you take that slide, you digitize it, and now instead of having each student come one after the other and then look at the slides, you can actually display that slide, right? You can display that slide, you can have everyone look at the same slide. So this is how it can actually help in teaching and training. So even if, for a second you say that maybe let's not use it for diagnosis, we can still use a traditional method, that's fine, but you can use these techniques for teaching, right? So that's why it can improve productivity. And a summary of how it can improve workflows, there's more innovations, and reduced turnaround times. So all of it results in increase in productivity of pathologists. Okay, so what is done? So you have a scanner, a whole slide scanner. You scan a complete microscope slide and create a single high resolution digital slide. And we'll see in a few minutes what this high resolution means. So you can capture many high resolution image tiles or strips, and then you can montage them to create a full image of a histological section. So the goal is to achieve efficient and cheaper diagnosis and prognosis and prediction of diseases. So yesterday we saw what diagnosis means, what prognosis means, and what treatment response prediction means. And all of this has been made possible by AI and the different machine learning tools that we are developing. So what are the benefits for patients? Let's say routine screening. It can help in enabling proper quality and safety of patient care, and increased efficiency and standardization. So what do I mean by standardization? So what I mean by that is, so let's say that you have, at site 1, you have your traditional ways of pathology analysis, and site 2 you have traditional way of pathology image analysis. A lot of times it becomes very subjective, right? So this results in some sort of loss in standardization. But now let's say that you have these slides that you can send to a center where it can be digitized, and then it can come back. So there's some level of standardization because the slides have been standardized under the same let's say digitization protocol, right? So there's some level of standardization that can be brought about by digital pathology. This is how a slide scanner looks like, right? So what happens is you have, let's say you have multiple different glass slides. The glass slides go in here, so you have the slots here, and once they go in here, they can be scanned. You have a high powered camera here, they are scanned, and then you see on your display how the image looks like, as opposed to having a microscope and putting that under the microscope and then looking at it. And there are multiple different companies that do this, Hamamatsu, Roche, and there are multiple different companies that can do this. Okay, so I hope I can play this video.
The number of patients in the UK that have to wait over 6 weeks for a diagnosis has increased 17% annually since 2010. This means that people like you and me have to wait much longer to find out if we have cancer or not. But why does the process take so long? Let's look at the current method. The process begins with a doctor taking a tissue biopsy from the patient. This sample is then sent to a pathology lab where it begins its preparation stage. The tissue is cut into thin sections, stained, and put onto glass slides. The sample is then referred to a histopathologist. That's someone who specializes in diagnosing diseases. They examine the sample and make a diagnosis. If they're unsure, the sample is packaged and sent to another histopathologist for a second opinion. The package could be sent to another town, another city, or even out of the country. This increases the waiting time by days or even weeks. This can result in unnecessary pressure and stress being added onto patients. Surely, with all the technology we have today, there must be a faster way of diagnosis. Luckily for us, there is a technology that could make a difference. Digital pathology. If we go back to where we started, instead of the tissue sample being packaged and sent away, the sample is digitally scanned and uploaded onto a secure server, where only verified pathologists can access it. The easy accessibility means that other pathologists can come together and collaborate in giving their opinions and suggestions. The pathologists can also examine the sample in great detail and make notes and highlight areas, aiding the communication between pathologists. Going digital can significantly reduce the diagnosis time. This means that patients can get their results much quicker than traditional methods and can begin treatment much earlier if necessary. So now, let's talk about the possibilities that digital pathology can support. With the current advancements of computing technology, pathologists could soon utilize the power of artificial intelligence, helping them to achieve a faster, more precise diagnosis. Artificial intelligence could help assist with identifying abnormal tissue or cancer, with quantifying the severity of disease, and it could help support a more personalized approach to treatment. This streamlined approach will improve diagnostic workflow for pathologists, improved support services for patients, better task prioritization, and helping assist pathologists in diagnosis. Why do we need digital pathology to be widely used across the NHS? In England alone, 13 million histopathology slides are examined each year. Since 2007, there has been a rising demand for diagnostic tests. This has coincided with a reduced pathology workforce capacity. A recent survey by the Royal College of Pathologists discovered that only 3% of pathology departments had enough staff to deal with this increasing demand. For the NHS, digital pathology will help meet these challenges and will provide patients with faster results and better care. Digital pathology, the next step in achieving improved diagnostic services and results for patients.
Okay, so this is
The number of
basically the summary of what I covered in the past few slides, and digital pathology is completely reshaping the way pathologists analyze images. So I was there at Tata Memorial a couple weeks ago. So they have an entire department that is dedicated to digital pathology. They have, I think, about 5 to 6 scanners, and all the slides that they get is digitized, and currently their pathologists are analyzing everything in a digital manner. So it will take time for other hospitals to catch up, but eventually, definitely in the next couple decades or so, the future will be completely digital. Okay, so this is not something new. This is not something that started 5 years ago or 10 years ago. The foundations of this were laid almost 50 to 60 years ago. And so there's an important lesson here, because we might think that a lot of these techniques that we are developing nowadays, yes, you write a paper and you forget about it, and you think that, okay, this probably will never see the light of the day. But that's not true. If you're diligent in doing whatever you're doing, and there's a significant unmet clinical need that you are addressing, eventually if there is need for that technique, it will show up in mainstream in the next 20, 30 years or so. Back in the day, I don't think no one even believed that technique like digital pathology could take off. But now you see how it's completely changing. The scene has completely changed. And obviously it's not, it did not happen just by itself. A lot of it was helped by the AI boom, right? You have a lot of compute infrastructure now, how you have different GPU servers, and processing of these images has become a lot faster. So all of that has aided digital pathology, right? So it all comes down to the fact that it's a collaborative thing, right? You can't just stand up a digital pathology unit by itself. You need the buy-in of the hospital IT, so that they can invest in compute infrastructure. You can have enough GPUs, you can have enough compute power that can support these kinds of activities. You need specialized hardware. It's not just that you take a camera and digitize it, right? You need specialized hardware. So like I said, the benefits, they include medical education. So telepathology is important because you might not have, so a lot of times you'll see that, for example, in India, you might not have specialized pathologists. So so you might not have specialized pathologists at all places, right? So there will be specialized pathologists somewhere else sitting remotely, but you would want to get their opinion. So let's say that there's a breast cancer slide that needs to be analyzed, and your hospital only has general pathologists who will be able to provide their interpretation, but in order to get something more specialized, it has to be sent to a specialized pathologist who specializes in breast cancer. Now, in order to do that, you can scan that image, you can digitize it, and you can just send it over to them and get their opinion, right? Or what you can do is you can open screen sharing, and then you can have a collaborative session where the specialized pathologist basically looks at the same region as you are looking, and they would annotate what regions are important, or if there's a particular disease that that tissue has. While they call what disease it has, they can tell you what parts of the image was responsible in detecting, that the patient has that disease, right? So you can see how it can enable collaboration, how it can enable telepathology. So clinical workflows, it can obviously improve clinical workflows, make it faster, and research. It definitely has played a huge role in developing AI-based biomarkers for many different diseases. So pathology education. So one of the ways of using digital pathology for education, like I said, is there are, let's say, a group of medical students who are doing a pathology rotation. So the pathologist would want them to understand that, okay, there's a particular image, and certain parts of the image has, let's say, certain diseases. So they can digitize it, put it up on the screen, and show them what the different parts of the image are. So, and it's interactive, right? So they can play around with the image. You can zoom in, you can zoom out, you can basically pan, and you can do all sorts of, you can maybe increase and decrease the brightness, or let's say that you apply some thresholding thresholding techniques to identify where there are particular structures. You can do that. So you can play around with the intensity of the image. So you can do all of these operations on your the software that you have. So if any of you is interested in looking at or playing around with digital pathology slides, you can access the TCGA website. So TCGA is an initiative in the US where they are collecting a lot of images and corresponding genomic data, and for a lot of these data sets that are out there, there are also corresponding radiological data on TCIA, the Cancer Imaging Archive. So this is a very rich resource. So I know that the last couple months there was something going on with the repository that we could not access many of these images, but I think they're probably back. But I'm sure if they're not there, they'll be there in a few months or so. So I think they have a collection. Last I checked, they had a collection of over 11,000 or 12,000 whole slide images, and this spanned multiple different cancers. So they have a very good interface. If you want to, let's say, look at only brain cancer, you can go there, you can build your own cohort that there's a particular cancer type you need. If there's some genomic data you are interested in, you can download that data, and you can also see if there's matched imaging data. So not all the cancers have imaging data. By imaging, I mean radiology data, but there are multiple of these cancers, especially lung cancer, which has, I think, rad path omics for about 150 studies or so. So yeah, I mean, don't quote me on the numbers. They keep changing. People keep adding their data sets. And also, let's say that here you are generating some data sets, you are generating some analysis results. You can upload your results to TCGA, right? You can I mean, obviously there needs to be, it's good if you have a corresponding publication. So one of the avenues that people have taken in the last few years is Nature Scientific Data. So that's a good journal where, let's say, you have some data sets that you have that you want to upload or you want to contribute to the scientific community. You have some analysis results. The data has to have some analysis results with it. Data cannot stand on its own. So you write a scientific data paper, and once it gets accepted, you can tell them, or you can obviously talk to the TCGA folks, and you can upload your analysis results. So let's say that there's some new feature that you've developed, some new biomarker that you've developed, and you want to compute this biomarker for each of the whole slide images or each of your radiology images that you have, and you want the community to use it. So eventually we want to do everything for the community to advance science, right? So if you think that there's merit, or you think that it will benefit researchers like you, or medical researchers, by all means you can approach TCGA, TCIA. They'll be very helpful, and you can upload your images, you can upload your omics data and your analysis results. So there is also another resource. I haven't noted it here, but I can write it down. Imaging Data Commons. So Imaging Data Commons is a relatively new initiative. I think it started in the last, it's been less than 5 to 6 years. So what you can do there is you can also upload your your scripts. And when you upload your scripts, they do provide some compute. It can directly interface with TCGA or TCIA. You can pull images, run it on their system. Right? So let's say that you have developed a new algorithm for, quantification of cells, or let's say you have developed a new algorithm. So this is mostly for radiology, for blood vessel analysis, like we saw yesterday. You can always upload your code. You can talk to the IDC folks. They can help you integrate it, and you can pull images that you haven't analyzed yet, and you can run it on their platform. So this is a relatively new initiative. This, I think, at Harvard. Yeah. Okay. So how else can we use digital pathology and AI for teaching, for education? We'll cover some of that tomorrow and day after. We'll cover generative AI tomorrow and day after. I think Dr. Beg also has a tutorial on that. So imagine that there's some kind of cancer for which you do not have enough images. You do not have enough whole slide. You do not have enough tissue samples, so you do not have enough whole slide images. So what we can do is we can use generative AI techniques like GANs or diffusion models. We can generate these these synthetic images, and then you can show these synthetic images, and obviously these synthetic images have to be blessed by pathologists so that it looks real. I'm sure you're ready with the question. Okay. So these images have to be blessed by pathology. I mean, these images have to look real, right? It's not just generating a synthetic image. It has to show proper characteristics of disease. If there's certain level of tumor infiltrating lymphocytes or certain level of cancerous tissue that has to be seen, then the generated image also has to show that degree of faithfulness. You can't just generate any image and say that because I have a prompt that says generate image of XYZ cancer, that it actually does that. You have to verify that with your pathologist. But once you have verified that with your pathologist, you can actually show that image to a medical student or to a resident or a fellow, and they can understand what these images look like. And also with generative AI, there's another fascinating aspect to it. So let's say that there's a certain disease, and this is a very hypothetical scenario, that for a certain disease there's certain layout, or the structure of the cells appear a certain way, the layout of the cells appear a certain way, the cells appear at a particular position. What you can do is you can actually, let's say you can extract the cells, you can segment the cells, and then you can train an AI model to give you similar images or images with similar cell layouts, but something else that's different, right? So you can essentially extrapolate your images to make it look like something else. So let's say that you have a particular grade of cancer, grade 1, grade 2, grade 3, grade 4. So a lot of times it happens in a continuum. It progresses from grade X to grade Y. So let's say that you have grade X, you have grade Z, and then you can ask your diffusion model or your generative model to interpolate what are the grades in between that, right? So that also helps you in teaching your residents, your fellows, as to how the disease progression happens. So there's the possibilities are almost endless, almost limitless, what you can do with generative AI. Also, I am not covering this in this lecture, but not all the slides are H&E stained. So H&E is one of the most common type of stains, which stains for the nuclei, the background, the epithelia, the stroma. But there are other kinds of stains that are more specialized, that have different kinds of stains for different kind of nuclei, right? So there are image-to-image translation methods, or there are generative methods where you can take one stain and convert it to another stain. So a lot of times, traditionally how this happens is, you have the tissue block, you section it, and so these are very thin sections. You take the first section, you stain it with one kind of stain, and you digitize it. Then you take the immediately next section, so that they are quite similar to each other. You stain it with another kind of stain. I mean, the stains are what? They're like proteins, right? They would they would attach to different regions of the tissue. So you would stain it and you would digitize it. Now, or another way of doing that is you take the first stain, after it has been digitized, you take that tissue that has been stained, you wash away the stain, and you restain it with another stain. So you can do that as well. I mean, sometimes it leads to loss in tissue, but that's something that's done routinely. But let's say that you have one stain and then you lose that slide for some reason, or you don't have the the sections that are around it. So you can take the available digital tissue or digitized tissue, and you can train your generative models to artificially convert it to another stain. That is assuming that you have enough paired data in your training, that you have stain A, stain B, you have enough data for stain A and stain B. You learn a function that can convert stain A to stain B with your training data, and in your testing phase, in your inference phase, you do not have stain B, you only have stain A, then you can use your model to convert stain A to stain B, right. So that's something that you can do using generative AI. And that's one of the hot areas of research nowadays. How do we artificially stain tissue with different kinds of stains? Okay, so teleconsultation, we covered that. So inter-observer and intra-observer variance. So this is very important, and this holds good not just for pathology, but also for radiology, or for that matter, any kind of image analysis. So because a lot of times what happens is, so today the pathologist would look at an image, and tomorrow they would come back, or let's say after a few days, they would come back, and for some reason they would want to change the diagnosis, or they have something in their mind that would sort of bug them and say that, okay, I want to go back, relook at my image, and have another look at my image and see if there's something that I have missed. Now that it's digitized, they would have, let's say, marked the problem areas, and they can immediately pull up that slide on the computer and go back to the problem areas and see if there's something that they missed and correct that, right. So that's basically intra-observer variance. So inter-observer variance. So you have two pathologists here. Pathologist 1 says something, pathologist 2 says something. Because you have this collaborative setup now with your digitized tissues, they can look at it at the same time, they can sit together and they can discuss and then arrive at a consensus if needed, right. So this can help assess this variance, or let's say you are conducting an experiment. So just another hypothetical scenario, you have come up with this really cool generative AI model where you are taking one kind of stain, converting it to another, or you are generating synthetic samples of a particular disease. Now you want the pathologist's opinion as to whether this synthetic sample that you have generated is good, bad, right? Good or bad or ugly or whatever, right? So you have then two different pathologists. You keep them separate, like you keep this study blinded. You provide the same generated image to both the pathologists, and you get their diagnosis, right? Not diagnosis, you get their interpretation as to how real that image is, how real that synthetic image is, or how real that fake image is. So you get their impression, and then you can compare them, right? So digital pathology can help you do that. Archiving of slides, we covered that. So digital reviews of diagnostic workflows. So this is again something very important. So you remember the last part of yesterday's lecture, we covered about eye gaze analysis, how we can take eye gaze. In fact, Moinak, who's sitting here, he worked on a lot of those projects. So we covered a few projects that we have been doing on eye gaze analysis. Now, what is done in a lot of these digital pathology software is, while the pathologist is looking at different regions of the slide, so I can try to illustrate that here. So this is your computer screen. So let's say you have the whole slide image here. Okay, I lost it. Okay, so you have the whole slide image here. So let's say the pathologist starts. Just a second. The pathologist starts from here, and then they move here, and then they move here, come here. So this is 1, 2, 3, 4. So just for simplicity's sake, let's say that you have these 4 regions that the pathologist traverses while they make their decisions. Now, what a lot of these software do is, they actually keep a record of where the pathologist is focusing their attention on, and they would have something like this that gets saved. This is the whole slide image. they would actually have this path saved, right. So, that is how the pathologist sort of moved around while looking at this image. So, this is nothing but connecting the centers of the fields of view, right. So, now what this can help you do is, so let us say that, so typically what happens in many of these clinical workflows is that junior radiologists or junior pathologists would make the decisions, they would make the diagnosis, and these have to be reviewed by senior pathologists. Now, if you had a provision like this where the scan path was saved, it was noted, then if there is any mistake while the senior pathologist is reviewing what the junior pathologist has done, they can tell them that this is the path they followed, or this is what went wrong when they were diagnosing this image. Does that make sense? Yeah. So, it can help provide digital, it can help in digital reviews of diagnostic workflows. And through this, you can also provide feedback to pathologists. So, remember, for the eye gaze analysis that we saw yesterday, one of the applications that we discussed was teaching, right. So, how if you can train a model to understand the eye gaze of specialists, you can use that to teach, you can in theory use that to teach a junior pathologist or a junior radiologist. So, by providing feedback, so if when they are looking at their images, if there is some portion of the image that they are missing or that they haven't really looked at, and if there is something interesting in there, your AI can tell you that, okay, here is an interesting region that you have missed, why don't you shift your gaze, go up here and then look at that image and see what is going on there. Okay. For clinical diagnosis, obviously it is important for clinical diagnosis. So, it has actually been shown, and this keeps getting better and better. If you compare the diagnosis that is made physically under the microscope and the diagnosis that is made using the digital pathology images, there is a pretty high concordance, and this is only getting better as pathologists are becoming more receptive to the idea of digital pathology. So, accuracy of reproduction of glass slide, it increases accuracy, and like I said, institutes have started switching completely to digital workflows. So, this is again very important, out of focus areas. So, I'll tell you why this is an important concern, and we'll discuss some of the techniques that we have developed in our lab tomorrow, but today I'll talk about what this problem area is. So, when you take an image on your smartphone, many times, I mean, smartphones are getting better, cameras are getting better, but let us say 5 years ago or 7 years ago, if you had, if you looked at some of these qualities of these images that were obtained by on your smartphones, you'd see that a lot of times because of, let us say, lighting conditions or subject movement, images would be blurred, right? And nowadays you do have deblurring algorithms on your, or deblurring let us say apps on your phone, but back in the day you didn't really have any deblurring apps. I mean, yes, you could back then you could upload them on MATLAB and do everything and do these things that you wanted, but from a consumer perspective, blurred regions was a problem, right? And not just that. So, this is something that you see on your smartphones, and we use smartphones on a day-to-day basis. So, this is a problem that has also crept into the medical workflows. So, we will see how this is an issue, not just in pathology, which we will see now, but also in radiology. Some of these applications, some of the algorithms that we have worked on, in fact Monak has worked on, is to deblur a lot of these images and make it diagnosis ready. So, in pathology this is especially important because these images are huge, right? And what might happen is, because let us say maybe tissue preparation issues or digitization issues, there might be certain parts of the image or the entire image that is blurred. Okay, so you have this whole slide image here, and for some reason you have certain parts of the image that are blurred. Okay. So, one of the ways, so what's the simplest way to fix it? Anyone? So, you have an image, so you basically have a whole slide image, you digitized it, and So let's say the tissue preparation is okay, there's nothing wrong with that. Something goes wrong in the digitization process and you see that some portions of the image are blurred. So what's the simplest fix?
Maybe you can pad them using nearest values, neighborhood pixels. You can pad them, mirror them, or copy them, and try to replace those pixels where it is blurred.
No, but that's a very, that's completely, it's wrong, right? Because if clinicians want to diagnose these images, you can't put some other information on there, right? It has to be something even more simple than that.
Is redoing it a possibility?
Redoing it, right? So one of the first things that comes to your mind is redoing it. So when our computers, for some reason you tried everything and it doesn't restart or something happens, you would reboot it completely, right? Or the old way of hitting your computer and making sure that it starts up, right? So the first thing that would come to your mind is, let's just redo it. But for some reason, digitizing it might not be possible, or let's say it will take time, right? So how do we solve that? We can think of developing and training AI models that can help us de-blur these images. So there are a lot of de-blurring algorithms that can take the out of focus regions, and based on the way they have been trained, so how are these models trained? So in order to train a deep learning model, one of the things, and there are multiple ways to do this, so one of the ways you can do this is, you can take an image, a high quality image, you blur it. So you have paired images. You have high quality image, the corresponding blurred image. You have multiple high quality images and the corresponding blurred images. So you have this paired data set, and you can learn a transformation, and I'm simplifying this by saying transformation, but you can actually train a deep learning model to understand what is the mapping between the blurred image and the de-blurred image, right? The blurred image and the high quality image. So now you can use this model to de-blur parts of the image, and now you will have a high quality image. And this image can then be used for diagnosis or for downstream AI applications. Okay. So another application similar to de-blurring or making these out of focus areas better is called super resolution. So we'll see in the next few slides how these digital pathology images are collected at multiple resolutions. Now I'll talk about that in more detail once we come to that, but just keep this in mind. So yesterday there were questions regarding site variance, right? So if there are images collected from one site, and then models trained with images collected on one side, and then you have your test data that is coming from another site, how do you ensure, what are the steps, what are the pre-processing steps that you take to ensure that your model works from images collected from another site? Right. So yesterday we spoke about a few methods for radiologies. Now for digital pathology, which I hinted at yesterday, one of the steps is called color normalization. So you have different color normalization techniques that you can apply, and back then you had non-deep learning based methods, but with GAN and with diffusion models, you have these generative methods that you can use to normalize the color intensity that helps you, analyze images that might not be in the same, let's say, color space. Right. So for example, here's an image of bladder cancer. Your machine learning models have been trained on images that have this kind of intensity. Right. Now what you do is, so your machine learning models, it's the other way around. Your ML models have been trained on images that have this kind of intensity, and now this is an image that you obtain, right, from another site, let's say. Now in order to infer this, your model using this image, what you would need to do is, you would need to color normalize this image to the intensity space of the first image, right? You don't have to do that. You can always take your model and try it out and see if it works, and a lot of times people don't do that, it still works. But a recommended pre-processing step is that you color normalize it to bring everything to the same space, and once you do that, you see how the intensity of that image changes. So this image is essentially image B that has been color normalized to A, and that gives you this C. Right? So B is color normalized to A, and it will give you C, and you see how these two, A and C, are now, they look similar in color space. Right? So that's something that that needs to be done before you can apply your deep learning methods. So even while training, so a lot of times you don't have data from, let's say, a single site for training, you have data from multiple sites for training. And we have seen this, we saw this yesterday where, I told you that for the COVID-19 case, when we trained a model on Stony Brook and tried to test it on data collected from Rutgers, it didn't work. And when we went back, looked at the X-ray images, we saw that the intensity profiles were different. Now, let's say that we want to train a model together. So we can obviously take Stony Brook data and Rutgers data, put them together and train a model. But even when we do that, we should make sure that to the best extent possible, the intensity profiles of both of them are brought to the same, let's say, space, and then we train a model, right? Now, what are the different kinds of applications that we can train AI models for? So segmentation. So similar to radiology, you'll have different regions where you have tumors, you have interesting tissue, non-tumor tissue, you have epithelia, you have stroma, you have different kinds of glands. So all of these primitives are important for pathologists to understand, right? So you can actually train deep learning models to detect and segment these important regions of interest. Like I said, you can train models to detect primitives like nuclei, like glands, and so on. And what is important is quantitative histomorphometry. So what does this stand for? So histomorphometry is nothing but quantifying the shape, size, and arrangement of histology primitives, right? So you can capture spatial features like orientation of nuclei, texture of nuclei, shape of nuclei, architecture of nuclei. So all of these are important measurements that can help you in downstream analysis of different regions of interest. Also, when you look at pathology reports, you will find terminologies, right, like pathologists will tell you these nuclei are rounded or these are elongated. Now, you can always give them an automated report that would tell them, okay, this percentage of the nuclei of interest are rounded or elongated, and they can always verify that with their own analysis, and then that's how you would validate your AI model. But the reason I'm telling you this is because a lot of these features, when pathologists look at them and they give you an assessment of the shape, the morphology of the different regions of interest or the different primitives, they are subjective, right? So they don't really give a number to that. But imagine if you had a simple, you had simple mathematical filters, or you had a simple, let's say, descriptor that can compute how round the nuclei is or how elongated the nuclei is. And you can assign a number to it, that this nuclei is 75% elongated, right? So think of it as, let's say you fit an ellipse. So all of you know about what an ellipse is. So you fit an ellipse, you look at the major axis length, the minor axis length, then you look at the ratio of the major axis, minor axis. So all of these can help you quantify the appearance of a nuclei. So think of it as if we are going from a more qualitative assessment to a quantitative assessment, and that obviously has important downstream meaning, because now you can tell the pathologist that this with certain degree of certainty that this is the shape of my nuclei. I'm not just saying that, okay, there is some roundness, some elongatedness, not that. I can give them an exact number, right? So that's something that AI can help us do. So just a simple illustration of the different ways we can, or the different possibilities that we have with the current AI tools that we are developing. So cancer localization, nuclei segmentation, epithelium segmentation, tubule segmentation. So we can basically segment out these different regions of interest that you see here. And then what can we do with the segmentations? We can extract the area, we can extract the density, the structure, texture, the spatial organization, the area, right? And then it's a lot of times you don't really need segmentation. It's okay if you can just detect the different primitives. You might want to, or the pathologist might want you to detect the where the lymphocytes are. So there are different kinds of lymphocytes, right? So these different kinds of lymphocytes mean something. So lymphocytes, like I said yesterday, they are the defense mechanism of the body. So and different kinds of lymphocytes have different contribution to the pathogenesis of the disease. So you can detect new lymphocytes, and what happens, what can you do once you detect the lymphocytes? You can actually quantify how the lymphocytes have infiltrated, like what is the infiltration level of the cancer nuclei with the lymphocytes and so on. And then mitosis, a lot of times these mitosis, so what is mitosis, by the way?
The division of the cell.
Yeah, division of the cells. So if you look at many of these slides, you'll see that there are regions where you observe the cell division. Now there are AI techniques that have been developed that can actually help you detect what portion or where that cell division is taking place. And if you think about it, many times it can be a needle in a haystack. So these images are huge. They can be 50,000 pixels by 50,000 pixels. So the images that on your phone are probably 4,000 pixels by 4,000 pixels or even lower, right? So these are huge images, and imagine a pathologist having to traverse, navigate that entire whole slide image just to find one mitosis. Now, if you can have an AI model that can, let's say, quickly go through the image and figure out where these mitosis regions are, that can essentially help reduce the time that it takes for a pathologist to study that image. Genetic subtype classification. So genetic classification is important because depending on what the subtype is, clinicians or oncologists can then decide what treatment needs to be given to the patient. So many times what happens is you have to send the tissue to a specialized lab to do sequencing and to identify what that subtype is. And this can be very expensive, this can be time consuming, and more importantly, because you don't have that, you may not even have that facility in your area. You might have to send it to somewhere else. It will take days and days to arrive. So, and this can result in the wait can result in a lot of trauma, and if you don't start treatment at the right time, this can prove fatal, right? So there's a lot of effort that's going on currently to just take the image and based on the image subtype decide what the molecular subtype of the disease is. So yesterday we saw in breast cancer, we saw the different subtypes: triple negative, HER2 positive, ER positive, right? So depending on what these subtypes are, oncologists prescribe different treatments. Now imagine if we can have an AI model that can just take the image and perform this subtyping and give you the results. So the doctors can look at these results, assuming that these are correct, or to a certain extent if this is correct with high certainty, then you can immediately start treatment and not have to wait for the actual the results of the genomic analysis to come back, right? And more importantly, this is not tissue destructive. You have that image and you do everything digitally. So when you do your omics analysis, you actually destroy parts of the tissue are destroyed. Now you don't have to destroy it. You can do this analysis, and then maybe after that you can send it to await confirmation from your wet lab, right? So that's something you can do. And then we are also using it for other higher order tasks like grading, prognosis. By that I mean we can take these images and try to train models to prognosticate how long the patient will survive, or if there will be recurrence of the disease or not, and so on. Any questions so far?
Sir, you told about the like whether we are performing the staining. The image to image, you told like suppose there is a one image, we digitized it, we stained it, and then we staining the second image based on the previous staining.
You mean digitize, stain it
digitize
digitally?
the first one, yeah, digitize the first one, and then second time we are digitizing it, we are taking the image, we are digitizing it and staining based on the algorithm, based on the deep learning method you told.
No, so what I said was you digitize, so you have stain 1, stain A, you digitize that glass slide that has stain A, and then let's say that you want to have another stain. Using your AI model, you translate the stained A image to stain B image.
Yes, so my question is, sir, the staining processes for different histopathological samples are different, like it based on the receptor and ligand mechanism.
Yes.
Mostly.
Yes.
So how you are going to like how it is that much efficient that we are using one stain to
Artificially
yeah,
generate enough.
artificially doing the another.
Yeah. So the only way this can be done, or let's say a reliable way in which this can be done, is if you have enough training data with both stains, right? So you have stain A for a tissue, and for the same tissue, or for immediately tissue right next to each other, the section next to that, you can have stain B. If you have enough training data for stain A and stain B for multiple different tissue, then you can confidently train an algorithm that can help you do the stain transfer. If you don't have enough data, it's difficult. I mean, there are unpaired generation techniques that people have been trying, but usually when you have paired data, this becomes a lot more feasible.
So in that case, we have to identify those stains
Yes.
which can be stained for both the digitized images, I guess. That will be more efficient. Like there is, suppose there are two type of stains, stain A and stain B.
Yeah.
And both can be used to digitize some specific portion of the histopathological sample.
Stains are not used to digitize.
So, to stain the specific histopathological sample, and then
Right.
we are digitizing it, and
Yes.
then we are getting the data.
Yes, yes, yes.
Thank you so much, sir.
So I just curious, in terms of when you are actually digitizing the slides, for a given slide, do they do multiple, like based what you have seen, do they do multiple like triplicates or replicates of the same slide multiple times, or just
For
once?
the same stain.
For the same, like
No,
let's
they
say
do
they
it
take
just
one.
once.
They do it just once.
Do it just once. But how it happens, so the internal mechanism of that is, it's not like, if you, so the tissue would be probably, I don't know, half of this, right? So let's say that this is the tissue, right? So if this is the tissue, you can't have that entire image taken in one shot. So what happens when the tissue is digitized is you break this up into tiles, and then small sections are sort of digitized and they're stitched together.
Okay.
And that's how they create the bigger image.
Matrix.
But they don't do multiple, they don't do it multiple times.
Okay.
Yeah. Well, okay, so let me take that back. Maybe I'm not sure about that, because if you think about it from the perspective of image acquisition, there might be some techniques where they take it multiple times and average it out, right? If let's say there is some error at the first acquisition, they might take it multiple times and average it out. But based on what I have read so far, it's just one time. Yeah.
So,
Yes.
can they build up a whole slide image on the basis of the image of different sections if they have?
Sorry.
So, suppose I have images for the different sections of a tissue. Can I use digital pathology to build up a whole slide image using the different sections of the image?
So you're saying you have a tissue section, and then what do you have?
I have images of the different sections of the tissue.
So when you have images of the different sections of the tissue, it's already digitized, right?
No, different sections of the tissues. I have slides for the different sections of the tissues.
Yes, slides. Okay.
But I don't have the slide for an entire tissue.
Okay.
So can I use digitize the different sections of the tissue and use it to build a whole slide image?
Okay. Yes. So you can do that. A lot of times it's actually done where. So you can read up papers on how prostate samples are digitized. So what was traditionally done is, if you don't have these glass slides where you can mount the entire prostate, they would divide it into 4 parts. They would essentially mount each of the quadrants on the whole slide image. They would digitize it, and then once you get the 4 quadrants, you would stitch them. So you would stitch them and get the entire prostate. So yes, theoretically you can do that with stitching mechanisms. Yes. And how do you stitch images? For those of you who have some image analysis background, I think you'll be knowing that. For others, so let's say that just to simplify it, you have one circle here, and you can't take the image of that entire circle at one go. So what you do is, you maybe take one portion first, and then you take the image of another portion. So you have to identify some landmarks. So this is one landmark, this is one landmark, and let's say this is one landmark, and you identify the same landmarks here, and then you stitch them. Okay. So, you have something called a homography matrix. So, you compute the homography matrix and then you multiply it basically and then with the image, and it brings that to the same coordinate space as the first one, right. So, that is how you stitch images. So, image stitching is a big field. So, in fact, all of you have it on your phone. So, what application on your phone camera has image stitching? Anyone? The panorama mode, right. So, when you have the panorama mode, you essentially what it does is, I mean, back in the day, maybe 5 years ago, the panorama mode was horrible. So, as you move it, you will see the size getting smaller or bigger. So, that is called perspective warping. Now it's getting a lot better with AI. So, what you do is, remember, if you notice it, if you go really fast, it will ask you to slow down. That is because it wants to take good quality images at each location, and then once it takes these images, it automatically stitches them and performs some other de-warping or unwarping operations, and it gives you a good quality panorama. So, yes, you can do it.
Sir, what kind of zoom level do we have to use, like if you wanted to train a deep learning model by using
good question, very good question. So, a lot of the foundation models that are out there, you'll see that these foundation models are trained at 20x. But so the reason they do that is one of the main reasons is computational efficiency. Like if we had theoretically infinite computing power, we would do everything at multiple resolutions, because you can, when you look at the way pathologists diagnose these images, they don't really look at it at 40x or 5x or 10x. They zoom in, zoom out, they look at it at multiple resolutions. So, ideally you would train a deep learning model or a foundation model at multiple resolutions and somehow combine these features to do your analysis. But because of computing constraints, a lot of these foundation models are trained at one resolution, either 10x or 20x. Very few foundation models are out there that have been trained at 40x.
Okay.
Yeah.
Like, does it changes with the different types of cancers like that we have to give?
It can, like it can. So, there are papers that have indicated that certain resolutions are better for diagnosing certain diseases and other resolutions are better for diagnosing other diseases. If you have some prior information like that, it is always good to start training your model with that level of resolution which is good for the particular disease.
Yeah, thank you, sir.
Yeah, okay. So, as a summary, what are the reasons why we want to use AI in pathology? First, time consuming and cumbersome tasks like manual annotations and cell counting, all of these can be reduced. But the primary idea is to have reproducible measures. So, if you tell, like with certain certainty or with conviction, that there are round nuclei or elongated nuclei in that particular tissue, then because you provide numbers, when you provide it to pathologist A, B, C, D, they see the exact same thing. Otherwise, what you have to tell them is very subjectively, very qualitatively, you tell them that, okay, you have approximately, half of the tissue is rounded or nuclei is rounded or something like that, which doesn't really help from a reproducible purpose. And there are these subtle image patterns that may not be discernible by human eyes, which can where AI can really help. And the ultimate objective is precision medicine by discovering new diagnostic and prognostic biomarkers. So, now let us come to what are some of the classical handcrafted features in digital pathology. So, yesterday we saw in the first part of the lecture certain radiomic features. You remember collage features which were used to study the morphology or the texture of the brain tumors. So, similarly, a lot of research has been done to study how handcrafted features in digital pathology can help in diagnosis, prognosis and treatment response assessment. So, here I would like to also tell you, all of you, that nowadays we are beginning to move away from handcrafted features. With deep learning, we've seen that it's much more powerful. The results are more accurate, better when it comes to things like tasks like detection of disease, diagnosis of disease, prognosis of disease. Typically, handcrafted features, even though those can be understood by experts, by pathologists, the performance of many of these features are not that good. But having said that, what we should understand is that let us not completely discount the handcrafted features, because these handcrafted features have an intrinsic meaning. right? And I'll tell you about what these handcrafted features are in the context of pathology. But what we need to keep in mind is that there has to be a way, or we should come up with ways to retain the high or the improved or the increased accuracy of deep learning methods, but at the same time not lose the interpretability that some of these handcrafted features can bring to the table. And we'll cover that in 2 lectures tomorrow. Okay, so by the way, the question that we want to address here is, can quantitative histomorphometry of the tumor morphology, can they predict disease aggressiveness and patient outcome? So what you see here are 2 samples of just part of the whole slide image, and here are the different kinds of features that we can extract. So we can construct graphs, and there are different ways of constructing graphs. You can have Delaunay triangulations, Voronoi methods, minimum spanning trees. So you can have graphs, you can have grayscale features. So grayscale features, by that I mean intensity based features. You can have texture features. And so there was this question about what magnification, right? So pathology images are collected at multiple different resolutions. So you go from 5x all the way to maybe 40x, 100x, 120x. So at different scales you observe different structures. So let's go back to the previous slide. Here you can see in detail where the different cells are. Now if you come out one level, you can begin to understand that cell that region of interest from the previous slide was probably somewhere here, right? So now you do see cells, but you also observe the context. So you see what are the different kinds of cells around it, right? So it's much more information you see as compared to just a small patch. We don't stop there. We can further zoom out. We can create graphs. We can look at the intensities, and then now you are beginning to see what the whole slide image looks like, right? So that small image that we saw in the beginning of the slide is probably one tiny speck here, right? So you see how we are getting more and more context, and this helps us. It's very similar to, let's say, what you have in your phones, the camera. The photos that you take using your camera on your phones. Even though the way of storage, the storage mechanism is different, you have a pyramidal format here, you do not have a pyramidal format there. You see, if you zoom in and zoom further into your images on your smartphones, you'll see it gets blurry, right? But if you zoom in or zoom out of the image that has been collected using a digital pathology scanner, it is not blurry. It will still maintain that level of resolution. It will still look, the resolution will still be very high because of the way these images are collected and stored. And then you can create, so the first slide we saw the graphs, those are more local graphs. These are more global graphs. So think of graphs as, let's say, connecting, the centroids of the different nuclei using some logic, right? So let's say that if the nuclei are round, then you connect them, all the round nuclei connected to each other, all the elongated nuclei connected to each other, you form a graph. So you can do that not just at a patch level, but you can slowly start to zoom out and construct that graph at a whole slide level, and this is what you see here. And so a lot of these measurements, these graph based measures can be used for downstream analysis. Further down you see how we now see the entire whole slide image. Okay, so now pathomics, so like we saw radiomics yesterday, so pathomics is nothing but the science of extracting features from digitized pathology images. So these features focus specifically on the section tissues, and the scanners, like we saw, they create the high resolution images that can then be digitally processed. So you can identify different kinds of cancer, you can annotate, you can look at multiple different resolutions. And what are the different features that you can extract? Shape based features, right? So shape based features, so this is the just a small tile from the whole slide image. You can extract features like shape based features where you are looking at how round the nuclei is, how elongated the nuclei is. You can look at the architecture. By architecture I mean how they are arranged with respect to each other. If you have one tissue type infiltrating into another tissue type, that means something, right? There are different levels of infiltration. All of this you can compute and you can assign a number. So a lot of these can be made quantitative. And then texture. So similar to what we saw yesterday with the Harlick features or the collage features, you can apply the exact same mechanism to these nuclei. So, once you segment out the nuclei, apply your Harlick features, apply your collage features and get the texture values, and this is how the the texture of the nuclei would look like, right. And why is this important? So, let's I hope you can see this on the screen. So, look at this particular nuclei here. You see how it's dark, and then look at another nuclei here. So, you see how it's a little bit brightish. So, when you compute the texture feature, you will see that the texture response of the nuclei here will be very different from the texture response of the nuclei here. And then you can actually use that texture response to maybe construct a feature vector for downstream classification analysis, right. Okay, so what are these different textures, and what do they quantify? So, a lot of times, because of different kinds of mutations, the texture of the nuclei might be different, or certain kind of texture might be associated with certain kinds of mutations. So, that is why it is important to study these textures. For example, in this particular case, you can see that the texture is different, and these kinds of texture led to good prognosis of the patient, and this texture led to bad prognosis or poor prognosis of the patient. So, what is the first step before most of this analysis? You have to segment out, you have to detect and segment where the nuclei are, and after you have segmented it, you can apply the different features. One of the very common features, like we discussed yesterday, is the Harlick features, and this is the entropy of the so Harlick has about 13 or 14 different features. One of the features is the entropy features, which quantifies the disorder of the pixel intensities, and if you compute that, you see how these how these responses are quite different. So, let's go to the other feature, shape feature. So, one of the simple shape based features that you can compute is nuclear size. And how would you compute the nuclear size? So, what you do is, first you have a deep learning method or some method to extract where the nuclei are, and then simply you compute the number of pixels. So, if you have a large nuclei, the number of pixels would be high, right, and if you have a small nuclei, the number of pixels would be low. And you see this is a much larger nuclei here, probably, I mean, it's part of it is cut off. So, you have a high number of pixels. So, simply counting the number of pixels within the segmented region within the nuclear contours can give you the size of the nuclei. So, solidity of the nuclei. So, this is an interesting feature where you want to quantify how solid the nuclei is. So, let us say that you have a nuclei that looks like this, and you have another nuclei that looks like this. The first thing that you do is you construct a convex hull. So, this convex hull would possibly look like something like this, and you construct a convex hull here, which would possibly look like this, right. So, you see that so what is solidity? So, solidity is nothing but proportion of the pixels in the convex hull that are also in the region, right. So, here is the nuclei, I am shading it here. So, clearly the proportion of this nuclei that is inside the convex hull is much lower as compared to this nuclei, right. So, the second nuclei that you see here is completely, it's very strongly associated with the convex hull, but the first nuclei you see a lot of gap between the nuclei and the convex hull. So, which means that the second nuclei is more solid as compared to the first nuclei, right, the first nucleus. So, these are simple ways of quantifying the shape of the different nuclei on the whole slide image. Now, another feature, like I was telling you, is axis ratio. So, you can fit an ellipse to the different nuclei that you have segmented, right. So, once you fit an ellipse, so let us say that this is a nuclei, you fit an ellipse to this, you have a minor axis, you have a major axis, and one of the very simple features can be the length of the major axis, the length of the minor axis, and the ratio of the major axis to the minor axis. So, these can be simple yet powerful descriptors for describing what the different nuclei in the images are. Okay. Now, going from shape based features to architecture based features, first what you do is, first of all, why are architecture based features important? So, the histological architecture has it's basically used, or you can use the architecture based features to quantify the structure of healthy tissue and the structure of diseased tissue. Now, if you have distinct structures, they usually indicate healthy tissue, and if you have loss in structure, it indicates uncontrolled growth or cancer. Now, if we compute graph based features, you see that for the healthy tissue, you have more well defined structures on the left as compared to the cancer tissue, where you have a different kind of loss in structure, right. So, the differences in structure can then be quantified and can be used to decide whether a tissue is cancerous or not. So, there are different kinds of architectural graphs that can be extracted. So, Delaunay triangulation, Voronoi diagram, minimum spanning tree. So, does not matter what algorithm you use. So, the first step of constructing these graphs is that you have to identify what the nodes are. Now, for the nodes of the graph, there are different ways you can do that. First, you segment out the nuclei. So, after you have segmented out the different nuclei, you can take the centroid of the nuclei and consider that as a node, or so that's a nuclei graph. So, let us say that you are interested in a gland graph. So, what you would do is you would segment out the glands in the image, and the centroid of the gland can be the node. So, depending on what kind of graphs you want to construct, you can segment out the primitives, identify the nodes, and then based on different rules that you have, you can construct the edges of the graphs. Okay. So, what else can we do with this? So, there is this notion of compartment aware analysis. So, it is not necessary that you have to analyze the entire tissue in one go. So, because these are huge images, there are different compartments in these images. You have epithelia, you have stroma, you have different kinds of cells, you have regions where you do not really have any tissue as altogether, right. So, you do not have any tissue in some of these regions for whatever reason. I mean, there can be tissue loss during the preparation, or for some reason there might be some hollow regions there. So, what we can do is you can have algorithms to separate out the different compartments and then do a compartment specific analysis, that this is what my stromal features look like, this is what my epithelial features look like, this is where you have the tumor infiltrating lymphocyte deposition, this is what my TIL features look like, this is what my healthy cell features look like, right. So, you can divide your image into different compartments and do a compartment specific analysis. So, this is the stroma, this is the epithelia, and you can see how the stromal cells and the epithelia cells have been color coded differently. So, you can do your analysis like this. So, different kinds of deep learning methods have been developed for analyzing these images. You can have methods like segmentation methods, like I said, where the method. So, one of the commonly used segmentation methods is called HoverNet, horizontal vertical network. So, this code is available and pretty widely used. So, this was published by Dr. Rajput's group from Warwick University. Since then, so this was the pre-transformer era. So, in the transformer era, you have methods like cell VIT. So, these are methods that are much more powerful, and they can not just segment out nuclei, they can also assign the nuclei into different classes. Right? So, you can have different classes of nuclei. Mitosis detection algorithms, like I said, cell counting. So, once you detect the cells, you can count the cells. But the challenge is that because these images are so huge, you would need large training data, and a lot of times, yes, you can develop these methods, but there is not much interpretability at the end of the day. But we'll see how we can address the issue of interpretability in tomorrow's lecture. So, tumor identification, classification, anatomical region classification. So, a lot of times the analysis focused on small patches because of computational challenges, but there are new methods like multiple instance learning. So, using multiple instance learning methods, we can take patches, smaller patches, and combine them to come up with a reasoning for the entire slide. Okay. So, maybe 10, 15 years ago, most of the papers that came out, you would see that the analysis was restricted to very small portions of the image. But now, we know that if we do that, we are losing so much information. There's so much available information in other regions, other parts of the image that we are losing. So there are methods like multiple instance learning, like I said, that can be used to come up with or arrive at decisions at a whole slide level rather than a patch level. So here are a couple different papers. So this paper that you see on the right, so there's this concept of super pixel or super voxel. So what does that mean? So you have, what you can do is you can divide you have pixels. So pixels are essentially each individual unit of the image, but you can look at pixels right next to each other. They share the same, let's say, similar intensity information, and the pixels that share the same intensity information, you can club them into one group. You call it a super pixel. So you take your image and you construct these super pixels. So you see this honeycomb like structures here. So these are all super pixels that you see here. So all the pixels inside the super pixel share some similar information, whether it is intensity or some, let's say orientation, entropy. So you construct these super pixels, and instead of doing your analysis on the pixels, you can do your analysis on the super pixels also. So that's something that can be done. So in this particular paper, the whole slide was partitioned into super pixels, and after that they did a graph clustering to group the anatomical regions, the pixels into the super pixels into different anatomical regions. And based on that, you can then run a classification to see if your super pixels belong to the cancer regions or the benign regions. Okay, so this is work from some colleagues from our group, Dr. Joel Salz, where the goal was to identify tumor infiltrating lymphocytes and then generate this spatial TIL map. So it has been shown in multiple papers that having good concentration of TILs leads to even good prognosis eventually. So that is why it becomes very important to identify where the TIL regions are. And once you identify where these TILs are or the lymphocytes are, you can essentially assign numbers to these patches. Okay, this patch has 98% TILs, this patch has 50% TILs. So you can construct a TIL based map, a TIL map, so which looks like, so this is a cancer map and this is a TIL map. So you see that here you have high concentration of TILs, here you have high concentration of TILs. And once you do that, you can provide this to a pathologist, and the pathologist can look at this and say, okay, this image looks like it has heavy TILs. This means that this person might have a good chance of prognosis if given a certain treatment, right? So you can perform this analysis, and in addition to showing the pathologist this image, you can also give them this image. Right. So yes, they can do that, they can look at TILs, but they can obviously not come up with a map like this immediately. But if you give them a map like this, they can look at the red regions, they can verify first of all whether it is correct or not. Assuming that the algorithm is well validated, they can look at your TIL map and they can tell you what sort of treatment, or they can tell the oncologist that this is the TIL map that we have for this particular patient, which means that this person would likely benefit from therapies like immunotherapy. So let's go ahead with that. I mean, the pathologist won't make this decision, the oncologist will, but the pathologist at least has the supporting information that we provide to them in addition to the digitized image. So this is work from our group, and this actually this was published 3 years ago at SPIE. So here we were looking at classifying images into 3 different groups. These were brain cancer images. So astrocytoma, oligodendroglioma, and glioblastoma. So we had the whole slide image. What we did was so this was before multiple instance learning. So we identified patches from it. We took features from each of these patches. So this was at a low magnification. So at a low magnification, we identified which of these patches would be important, and then looked at only those patches at a high magnification to do the analysis. So that's what we did in this paper. But nowadays, we have moved on and we look at whatever magnification we want to, but look at the entire image. We don't pre-select important patches. That is what multiple instance learning actually gives us. It gives us an attention weight for each patch. It tells us which patch was eventually important for the downstream classification. You don't have to pre-select patches. So why else or where else can we use these digitized pathology images for multimodal integration with other cancer. So, so there's a lot of research that has happened in RAD path registration. So here's an example of a prostate scan. This is a radiology scan of the prostate, and this is the corresponding tissue section of the entire prostate. So there was this question that was asked, what if you have different sections, can you put them together? So this is what I meant when I was explaining to you the different quadrants. So previously, if you did not, like when we did not have the capability to digitize the entire prostate, they would actually take these 4 sections, digitize them separately, and then stitch them to get this entire prostate. So now that you have the prostate sample, digitized prostate sample, you can also, let's say that you have the image of the prostate that was taken before the prostate was removed from the body. Now you can have a registration method that you can use to register the MRI onto the pathology or the other way around. Now what this can tell you is which part of, so let's say that you had this part of the image as cancer, that would correspond to this part. Now you can study what is the radiology characteristics, what are the radiology features associated with this part, and what are the corresponding pathology features in this part. So once you understand, or once you have the features coming from different modalities, you can do some sort of a correlation analysis, associative study to see how features at one length scale are related to features at another length scale. So there was this question yesterday, like, okay, how do you even understand what's happening, what's the underlying biology? So you get something in the radiology length scale, what's happening in the pathology length scale. If you have mechanisms like this, where you can co-register images or radiology images onto pathology images, then you can identify the exact same area where where tissue are corresponding to each other and study them in great detail. So not just images. So similar to radiology, pathology images also have reports, right? So we can extract reports, and then we can use the reports to do multiple different things. So this slide that you see here will come back to this tomorrow. But what I want to say here is, what we did here in this particular project was, we took the images, we extracted different kinds of features like TIL based features, tumor based features, but we also took the corresponding pathology report, we provided it to a large language model, and in this case it was GPT. We provided so the model gave us a summary of what the report had. So okay, why did we want a summary? So I don't think we'll go into the details of this, but we wanted a more concise representation of the report. Like, the report will be long, but we did not really want a long report. We wanted something short and a concise report. So once we had this concise report, we provided it to a language embedder. We got the embeddings, and then we had the image-based patches that we got. We got the embeddings from that, and then we used this conditioning mechanism to a model, to a generative model, to help us generate synthetic samples of that image. So we'll come back to this and discuss this in more detail tomorrow. So, but the bottom line is, what I want you to take away from this is, there's multiple modalities of information within this. So we have digitized pathology slides. You can have different kinds of stains, like we discussed. It's not just H&E stain. You can have DAPI stain. You have Key 67 stain, different kinds of stain, and then also you have the pathology reports associated with those slides. So it's not just imaging and pathology. You can also combine genomics. Obviously, you have genomics information corresponding to those. You can have proteomic information. You can have immunohistochemical features, and you can combine all of this information theoretically to get better classification, prognosis prediction, and treatment response assessment. So ultimately, what we want to do is, we want to combine histomorphometry with molecular data for more holistic disease characterization, and that would lead us a step closer to precision medicine, which is essentially tailoring a treatment plan to each of the individual patients that we have, using data from multiple different sources. So what are the different challenges? Obviously, interpretability. So like I said, a lot of times we are beginning to move into an era that is completely deep learning dominated, which is important because we have a lot of computing power, and we have seen that if we can apply these deep learning techniques, the accuracy of these different classifications increase. But it comes with a risk of decrease in interpretability. A lot of times interpretability doesn't really matter. For example, if the goal is to just segment the nuclei, right, just to segment the glands. If your model does well, your model does well. We don't really care about how that model works, or the doctor won't really care about how the model works, as long as you say that, as long as your model gives you the right regions of interest. But if it comes to treatment response prediction, and your model tells you that this patient will survive for n number of years and this other patient will survive for p number of years, if n is much lesser than p, right? So then the doctor will want to know, why did the model sort of give you this prediction that this patient will survive for fewer number of years as compared to the other patient. So that is where interpretability becomes crucial, right? Color variations, we covered that, so that can lead to issues in training and testing, and we need to make sure that we do our due diligence to ensure that this color variation is accounted for in your model training and inference. And so these slides that you saw here, they're all 2D in nature. So yesterday the 3D images, the MRI images that we saw, they're actually 3D in nature, right? So you have the brain, you take the MRI of the different sections, so it's a 3D image intrinsically. So even though the tissue that you extract from these histology images, they are 3D tissue, because it's not just one slice, right? You'll extract a bulk of it. But when you section them, these essentially become 2D sections. Now there is a lot of research now going on in 3D pathology. So you can actually take the 2D sections and stitch them together to form a 3D histology, digitized histology image. Now what it can help us do, so if you reconstruct or if you stitch these images together or layer these images together appropriately to get your 3D histology sample, you can actually look at structures. So there will be blood vessels, or there will be some other structures which you can start to observe in 3D, right? So we are moving from, there's a lot of research that's going on where we are moving from 2D to 3D analysis to get the depth information. Multispectral images, it's not just a single image, but there can be multispectral images. And last but not the least, we need high performance computing because of the high data. So this is truly big data. So if you look at one whole slide image, it can be anywhere between 1 gigabyte to maybe 3 to 4 gigabytes. So these are huge images, and a lot of times it's very impractical or almost impractical to load the entire image into your RAM and do the processing, right? Because you'll have to, it's not just one image, you'll have to do them in a batch, you'll have multiple different images. So there are methods that people are developing to see how these images can be loaded onto your computer memory in order to make the processing more efficient. Okay, so I think that's it for the first lecture of today. Any questions?
What are multispectral images?
So by multispectral, what I mean is, it's similar to if you have multiple different channels, right? So let's say that you have one stain and another stain, you can look at different channels of the same stain, and this basically become multispectral.
Sir, we have seen that we are using now deep learning techniques to extract features and automatically classify images.
Yes.
And sometimes you mentioned hand-captured features are not useful. So I have in mind, like in some applications where two different classes have similar textures and everything,
yes,
and the model fails to classify, there is no clear-cut boundary. So how can we combine efficiently this along with deep learning and handcrafted features? Is there any way?
Yeah, we'll see tomorrow in tomorrow's lecture. But I can give you a teaser. So let's say that you have a deep learning model and you have your handcrafted feature model. So what we can do is we can train a deep learning method to learn from both handcrafted features and the actual images. So if we come up with a co-training pipeline, we can basically use the high, we can leverage the high accuracy of the deep learning methods, but at the same time retain some interpretability coming from the handcrafted images. But we'll see that architecture tomorrow. Yeah. And it's not just handcrafted features, right? I mean, yes, handcrafted features are, end of the day, handcrafted features are something that we craft as as scientists for the pathologists. But what is probably more important for them is if we can use some language-based features, right? So what if you can say that, okay, there is in exactly this in this terms, you can have a sentence saying that, okay, X nuclei or tumor infiltrating lymph, there's a heavy infiltration of cancer cells into the tail region or the other way around. So these kinds of descriptions are better understood by the pathologist. So if we can train a model, a deep learning model that can take into account these descriptions and use these images, and when you provide the output, you can say, okay, this is a cancer because of this concept. That will be more important. So that's also something we'll see tomorrow.